Hello everyone. So today we will be talking about activated methyl cycle and methionine, or it is also called methionine cycle or methionine cycle. So this is the third part in series of sulfur containing amino acid metabolism and homocystinuria. So let's talk with the today's topic. So if you have not seen the previous two videos, please go and watch this previous two videos so that it will be helpful for you to understand this video also. And let's begin with the today's topic. So why do we need to understand this methionine cycle? So we need to understand this methionine cycle or activated methyl cycle to understand this pathology. First is homocystinuria, especially type 2 and type 3, megaloblastic anemia, and then along with megaloblastic anemia, folate trap. So these three core concept, understanding of this three core concept will require understanding of this activated methyl cycle. Now let's first talk about this methionine, activated methionine. Activated methionine or activated methionine is also called SAM, S-adenosyl methionine, S-adenosyl methionine. So this S-adenosyl methionine is this methionine amino acid. This is methionine amino acid. This methionine amino acid with its sulfur group in methionine is attached to this adenosine. Adenine plus ribose sugar, adenosine. So this is attached to adenosine. This is how, because of this methionine gets activated. If you can see here, so in this picture only, if you can see here, the sulfur is bond with 3 carbon, and that makes this sulfur very active group and very active to donate this methyl group. And because of this 3 bond, and there will be positive charge, and this positive charge enables this methionine to transfer this methyl group very easily. And in other words, this type of compound, this SAM, has very high transfer potential. This is called transfer potential. So very high transfer potential. Transfer potential means very high capacity to transfer this methyl group. So transfer potential of methyl group is very high for this compound. That is the significance. That is why this is called activated methyl cycle. So let's talk about the cycle itself. So see this sulfonium ion. I was missing one point. Because of this plus charge, this becomes sulfonium ion, and this sulfonium ion is the heart of this molecule. Because of this only, the transfer potential of this methyl group is very high. This methyl is active. Now, so let me talk about this also. So ATP, this activated SAM, is synthesized from ATP. Because this is ATP, so ATP, when it's bind to it, so it is activated, and that is why this is called activated methionine also. That is why this is called activated methionine also. So this important concept is required to understand this activated methyl cycle. Now let's talk about this concept. So what happens is methionine will convert into S-adenosyl methionine. So this is methionine. The upper part is, this is methionine. This amino acid is methionine, and this methionine is a sulfur containing amino acid that has about 6 carbon, if you can see, and this last methyl group is bridged with sulfur. This is called thioether bond. Now this methionine will convert into S-adenosyl methionine, and this S-adenosyl methionine, after donating this methyl group to some compounds for synthesis of, so methyl group will be donated for synthesis of epinephrine from norepinephrine, catecholamine. This is a catecholamines, then creatine synthesis and other molecules that I have already talked. You can see the previous video. And then so after transferring methyl group, this S-adenosyl methionine will convert into S-adenosyl homocysteine, and this S-adenosyl homocysteine now will separate. Adenine and homocysteine will separate now, and then this will form homocysteine, and adenine, because of hydrolysis, homocysteine will be separated. Cysteine will be separated, and this again homocysteine will be recycled to form to form methionine. And this recycling requires methyl group. But this methyl group, this homocysteine gets this methyl group from tetrahydrofolate, methyl tetrahydrofolate. This methyl group, this homocysteine gets this methyl group from methyl tetrahydrofolate, and this is a cycle. So again methionine will be formed. The same, what will happen? So activated methionine, that is SAM, will be formed, and SAM will donate methyl group to form certain compounds, and then it again will convert into S-adenosyl homocysteine, and then homocysteine will be like this. Homocysteine again will be regenerated or recycled, and this cycle goes and goes in the cell to actually preserve this methionine. Methionine is a essential amino acid. So this is called activated methionine cycle. Now let's see this reaction. The same thing here, you can see the reaction. So homocysteine is being, methionine is being converted into, this is methionine, and methionine is being converted into molecule called homocysteine, and this homocysteine again will convert back to methionine, and this is one cycle: methionine, homocysteine, homocysteine, methionine, methionine, homocysteine, homocysteine, methionine, and this is called activated methyl cycle or methionine cycle. Why it is called activated methyl cycle? Because this methionine converts into one very important compound, activated methionine, as I've already talked about. This has very high transfer potential methyl group, and this methyl group will be donated to form certain compounds, catecholamines, creatines, and then some methylation for genetic regulation, formation of choline. This all reaction requires methyl transfer, and this methyl is transferred from very activated methionine SAM, and this reaction and after catalyzing this reaction, SAM will convert into S-adenosyl homocysteine, and this S-adenosyl homocysteine will lyse through the enzyme called S-adenyl homocysteine hydrolase. The water will be required to break the bond between adenine and then homocysteine, and this homocysteine will be generated. Now homocysteine will be regenerated in single step. You can see the homocysteine formation from methionine, it requires 3 steps. So in between, very important step is methyl transfer. This is very important function. The function of whole cycle is the methyl transfer. The function of whole cycle is to transfer methyl to certain group. This is the whole main function of this cycle. Now the first reaction is done by MAT enzyme, methionine adenosyl transferase. This MAT enzyme, you can remember MAT enzyme. Second is different methyl transferases are there for different compounds. For example, in catecholamine synthesis, formation of epinephrine from norepinephrine require PAN-MT and PAN-MT. So this is name of long enzyme you can get. So this, the full form of this enzyme is actually phenylethanolamine and methyl transferase. Now this enzyme, and for other there will be other, but these are all methyl transferases. Different type of enzyme methyl transferases will be different reaction, but they are all methyl transferase. And then third reaction is always S-adenosyl homocysteine hydrolase, and this will produce homocysteine, and this reaction will be catalyzed by one enzyme called methionine synthase. The name itself suggests methionine is being synthesized, that is why methionine synthase. And second name of this, the same enzyme is called, the second name of the same enzyme, methionine synthase. Methionine synthase is also called homocysteine methyl transferase. Homocysteine, homocysteine methyl transferase, HMT. So methionine synthase is homocysteine methyl transferase. These are the name of same enzyme. Let's see what happens next. So this part of reaction requires 2 vitamins also, folate and vitamin B12. Let's see how does it happens. Now methionine synthase will catalyze this reaction. This methionine synthase has this vitamin B12 is very integral part of methionine synthase. So vitamin B12, especially methyl B12, methyl cobalamin, especially methyl cobalamin. So methyl cobalamin will convert into cobalamin. So we through donation of methyl group to this, and this where does this vitamin B12 get methyl group? It gets methyl group from methyl tetrahydrofolate. So this is how methionine synthesis utilizes this methyl group as well as this folate as well as this uh, folate as well as this vitamin B12, folate as well as vitamin B12. Now where does this this tetrahydrofolate comes from? So tetrahydrofolate actually comes from amino acid serine. And then serine will convert this tetrahydrofolate to methylene tetrahydrofolate, and this methylene tetrahydrofolate will reduce to methyl tetrahydrofolate. So this methyl tetrahydrofolate, ultimately this methyl group here is coming from folate and this from amino acid, and this is the whole reaction of this activated methyl cycle or methionine cycle. You can see at this side on the left there is methionine synthesis. On the left you can see short of methionine breakdown reaction. Sorry, methionine breakdown reaction is there. So in right side, left side there is methionine synthesis, and right side there is methionine breakdown and methyl transfer reaction is happening. Just it's very easy cycle but very important cycle. What happens now? What is the significance then? So the significance of this cycle is this enzyme. Generally, this enzyme is deficient in type 3 homocystinuria. When this enzyme is deficient, this will cause type 3 homocystinuria. What will happen? So if this enzyme is deficient, there will not be production of methyl tetrahydrofolate, and if there is no production of methyl tetrahydrofolate, so there will not be reactivation of this methionine methyl, this cobalamin present in methionine synthesis. It will the cobalamin will not be converted into methyl cobalamin, and if it is not converted into methyl cobalamin, so this homocysteine will not be rejuvenated. If it is not rejuvenated, so homocysteine concentration will rise, but methionine concentration is drop. So this is the significance. So in type 3 and type 3 there will be high homocysteine and low methionine. Type 3 there will be high homocysteine and low methionine, and this is because of generally the most common defect is this enzyme. This enzyme is called methylene, MTH means methylene, MTHF, methylene tetrahydrofolate reductase. So this requires NADPH, and if this enzyme is absent, so this will happen. So no methylation, and that is why high homocysteine and then low methionine. So and then second type of homocystinuria is because of type 2 homocystinuria. So type 2 homocystinuria is because of deficiency of vitamin B12 methyl, this vitamin B12 metabolism. There is problem in vitamin B12 metabolism. Actually vitamin B12 metabolism, intracellular metabolism, and incorporation of this vitamin B12 into this methionine synthesis require a series of protein. So protein are like cobalamin, so cobalamin protein, CBL proteins. So cobalamin protein are required. There are C, D, E, and these proteins are required. So generally C proteins are deficient in this methyl cobalamin metabolism. Generally this C protein deficiency will cause type 2 homocystinuria. So type 2 homocystinuria, if vitamin B12 is not there, if this enzyme is defective or vitamin B12 is not there, what will happen? So there will be high amount of folate. So folate, there will be tetrahydrofolate, but this tetrahydrofolate does not get the way to be incorporated into this homocysteine, and that is why this when there is defect in vitamin B12, there will be high accumulation of this tetrahydrofolate. So vitamin B12 deficiency will cause high methyl tetrahydrofolate, and then not only this methyl tetrahydrofolate, same thing will happen here also. So there will be high homocysteine and then low methionine, high homocysteine and low methionine. So this vitamin B12 has one big phenomenon. This phenomenon is called folate trap. though it is very high, but it is not get reactivated, so it is not being converted into tetrahydrofolate so that it will be utilized further. And that is why this whole mechanism of trap of this folate is called folate trap. And this folate trap is very important. Folate trap. Folate trap. Why this is very important? Because this folate trap is responsible for megaloblastic anemia in vitamin B12 deficiency. Now I'll repeat, folate trap is responsible for vitamin B12 deficiency, while folate trap is responsible for megaloblastic anemia and vitamin B12 deficiency. Folate trap is responsible for megaloblastic anemia in B12 deficiency. So this is very important one. So we have talked about folate trap. What is folate trap? And then we have already talked about type 2 homocystinuria and type 3 homocystinuria. So what happens in type 3? Let me revise. What happens in type 3 homocystinuria? Type 3 homocystinuria is because of folate metabolism defect, and generally the most common enzyme deficiency is methyl tetrahydrofolate reductase, and because of that what will happen ultimately is this homocysteine will not convert into methionine because of lack of transfer of methyl group, and then as a result homocysteine will increase and then methionine concentration will decrease. Now type 2 homocystinuria is because of deficiency problem in vitamin B12 metabolism. Because of that this methionine synthesis will not have its methyl cobalamin, and because of deficiency of this, so especially generally deficiency is cobalamin C protein deficiency. Because of this abnormality in vitamin B12 metabolism, it will cause type 2 homocystinuria. And type 2 homocystinuria has additional one finding is like folate trap. So folate will also be high. Here folate will be, methyl tetrahydrofolate will not be high, methyl tetrahydrofolate will be low, but in this case methyl tetrahydrofolate will be very high. So this is called folate trap. And in vitamin B12 deficiency, so cause of megaloblastic anemia is folate trap. So remember, these are the important point of this point about this activated methyl cycle and methionine cycle. So thank you. And then let me talk about this last concept about this. So last concept, it is very important for treatment purpose of this. This last concept is very important for treatment of the this disease. So last concept is, if you can see the change in slide. So methyl. So this whole reaction can be done by direct transfer of methyl group from this compound called betaine. This compound called betaine. So when there is defect in this metabolism of vitamin B12 and this, so we give the treatment as betaine. Treatment is given, betaine is given in treatment. So betaine is given so that this conversion of this homocysteine into methionine can happen. So this enzyme can use this betaine also. So no, sorry. So there is different enzyme for this conversion of this homocysteine into methionine, that utilizes betaine, and that is why this is very important concept for treatment. So in defect in this condition, we have option of betaine. If we supplement with betaine, so this can be, this homocysteine will be rejuvenated, and because of rejuvenation of homocysteine methionine, so this cycle goes and goes and on, and we will not have problem. So this is all about this concept. We have already talked about type 2 homocystinuria, type 3 homocystinuria. We talked about folate trap, and fourth point we talked about this betaine as option for treatment. Thank you for watching this video, and if you like this, if this video helped you, please like this video and subscribe the channel. Thank you.